The potential for antisense oligonucleotides, or ASOs for short, in treating neurological disorders has increased rapidly due to the success of ASOs in treating both Duchenne's muscular dystrophy and spinal muscular atrophy. However, before these ASOs can be tested in clinical trials, it is essential first to have a recapitulating animal model where you can test both the efficacy of the ASO as well as any potential adverse effects. Today, I will be sharing with you the work I have done in modeling and characterizing a FUS R536H point mutation in zebrafish. This point mutation is orthologous to the FUS R521H point mutation seen in ALS patients.
So what is ALS? Amyotrophic lateral sclerosis is a neurodegenerative disease that results in death of motor neurons. It was founded and named by Jean-Martin Charcot, the gentleman on the right. The main cause of death is respiratory failure. 90% of cases are sporadic and 10% are familial. 5% of the familial cases are due to mutations in the fused in sarcoma gene, which encodes the FUS protein and is my protein of interest. The bar graph on the left displays the onset of FUS ALS cases and shows the most common variant is at the R521 position. But what does FUS actually do? As a DNA-RNA binding protein, it sort of acts as a jack of all trades and is involved with DNA repair, transcription, alternative splicing, and RNA transport. Using CRISPR-Cas9, our lab has developed a FUS model of the analogous mutation encoding the FUS R521H variant in the zebrafish genome, encoding the FUS R536H variant, as well as a FUS knockout model by also using CRISPR-Cas9 to target the ATG start codon in FUS. Here I have pictures from wild-type homozygotes and knockouts from 1, 2, and 3 years of age. I want to point out this interesting curvature of the trunk we see in our 2 and 3-year-old homozygotes, which is absent in our other lines. This is a graph of the ongoing adult survival curves I've been working on. We will see that both the heterozygotes and homozygotes have already reached median survival. However, this is not the case for both the wild types and the knockouts. So first, I want to briefly tell you about the larval locomotor free swim experiments I've conducted in both 5 and 10 days post-fertilization. At 5 DPF, my knockouts have significant increase in locomotion in comparison to heterozygotes. At 10 DPF, the wild-type larvae had significant increase in locomotion in comparison to both the heterozygotes and knockouts. Next, I'm showing the locomotor characterization I conducted in my adult models at 1, 2, and 3 years of age. The top right shows heat maps from different 1 and 3-year-old wild types, homozygotes, and knockouts. It is clear that the homozygotes at 3 years of age have reduced locomotion in comparison to the other 3-year-olds. When looking at the mean velocity in these free swim assays, on the left, we see a significant difference between the wild types and homozygotes at 3 years of age. Clearly, there is a reduced locomotion in the homozygotes, but why? To address this question, I've begun imaging spinal cord sections with DAPI and CHAT, a marker for motor neurons. These sections are taken from between the 2nd and 4th vertebrae of the spinal cord of 3-year-olds. We only see 3 motor neurons in the homozygotes R536H slides in comparison to 6 in the wild type slides. I've recently performed RNA sequencing on 1-year-old spinal cords from wild types, homozygotes, and knockouts. Here I have Venn diagrams displaying the number of significantly up and down regulated RNA transcripts between both the homozygotes and the knockouts. And we see that there is a vast increase in the number of differentially expressed transcripts in the R536H homozygotes. To further illustrate this, I generated volcano plots, and it is obvious that the change in significantly up and down regulated transcripts in the homozygotes versus the knockouts.
Finally, I'd like to bring you back to what I started this presentation with, and that is the use of ASOs as potential therapeutics for neurological diseases. A new experimental ASO called JACIFUSEN is being prepared in a clinical trial with 8 ALS FUS patients. JACIFUSEN is named after Jaci Hermsted, who was diagnosed with FUS-associated ALS on February 14, 2019. Jaci received her first treatment on June 11th, and after 2 months, she returned home. Sadly, Jaci passed away on May 1st, 2020. Details of the effect JACIFUSEN had on her ALS progression have not been released. However, in the case of Jaci, it shows the need for testing this ASO in a recapitulating ALS animal model, so we can determine its efficacy, the optimal concentration, and its timeline. Although not complete, my R526H zebrafish line shows promise as a successful recapitulating model, and as such, I'll begin testing ASOs on my lines in the future. With that, I would like to thank my lab, the Armstrong Lab, my advisory committee, and the Rare Neurological Disease Group, and of course my funding sources, CIHR, ALS Canada, and HBHL. Thank you so much.